ProfsCast with Professor Fensterle and guests. Yeah, welcome to the ProfsCast. I'm Joe Fensterle from the Rhine-Waal University in Kleve in Germany. And today my guest is Patric Bourceau. Patric, welcome to the ProfsCast.
Yeah, thanks for having me.
Yeah, so as you see, we're sitting in the outside today. It's 34 degrees Celsius, I think. So we decided we cannot go inside, but we're at a special location. We are here at the Rhine-Waal University on the campus of the Rhine-Waal University. And therefore, we do the interview here. And what you will see later on, Patric is a very special guest because he's a former student, more precisely, he's a first-year student of bioengineering, which makes me happy. And as he's a student, of course, a former student, we also have as a motivation here a beer that the interview gets smoother at these 34 degrees. Patric, I also already said that you're a former student and we hear. So if I say you're now an electron transport guy, is that completely wrong? Or could
That's
you
not completely wrong. So the transport of electrons to generate energy, which can then be used to fix carbon, for example, is the key feature of the symbiosis we're working on. So we're working on chemosynthetic symbiosis in my department, where invertebrate animals live together with bacteria in regions where they have no access to other food sources. For example, the mussels we're working on, Bacillus mussels, they live in the deep sea where they don't have any algae where they can filter feed on. So they completely changed their body plan, their gills, their respiratory organ is now filled with bacteria. Those bacteria can tap into chemical energy sources like methane or hydrogen sulfide, which comes out of the seafloor in these black smokers or hydrocarbon seeps. And they share the energy they can generate from oxidizing those substrates with their host. In return, they get shelter and always a fresh stream of new nutrients provided by the mussels.
Okay, we will come more precisely on the research later. So at the end, you work on symbiotic relationships between bacteria and vertebrates, to summarize it briefly. But where do you actually work at present?
Right now, I'm doing my PhD at the Max Planck Institute for Marine Microbiology in Bremen, in the Department of Symbiosis in the research group of metabolic interactions.
Okay, so now this is what you do now, but your scientific career, I know at least one part of your scientific career which started here at the Rhine-Waal University. Could you briefly state where you have been already?
So yeah, I did my bachelor's degree here, and then for my internship I went over to the Netherlands, just 15 miles that way. In the Department of Microbiology.
At the University of Nijmegen?
Yeah, at the Radboud. And my key into this was basically that I knew how to work with bioreactors. So that was my entry into the world of basic research, I would say. And then I stayed there for my bachelor thesis also. Then I was intending to also continue in the Netherlands. But then I got the offer or the invitation to join my former boss to come with him to Bremen because he was building his own research group there. So I worked 1 year for him, and then I started the MARMIC program. That's the International Max Planck Research School for Marine Microbiology at our institute. So it's a combined program for master's and PhD candidates. And I did this master's, which is a fast track, so 1.5 years for 120 credit points. And then I stayed there to do my PhD.
Okay, so the first step was you had a professor who showed you to work with bioreactors.
That's where it all started, yes.
That was the starting point. That's nice to hear. So we don't talk about who the professor was, but he was here at the Rhine-Waal University.
He was here, yes.
You went to the Max Planck. That's interesting. So directly after your bachelor thesis, you went to the Max Planck Institute. Did they pay you or?
Well, yeah, first I wasn't studying for a year. I was working, so I was paid for my work. Yes.
So you started as right after your bachelor here in bioengineering at the Rhine-Waal University. You started as a staff scientist at the Max Planck Institute.
I don't know if staff scientist is the correct word. I don't even know what my title was back then. My job was basically to to build up the lab. So it was an empty lab basically, which we joined, and then we ordered the bioreactors. I had to arrange for like the gas lines to be set, order a lot of stuff, set up the first reactor, set up the GC, develop protocols, at one point write a safety concept for nitrous oxide, stuff like that.
Yeah, so this is what we would term is usually a work of a postdoc. So this is a staff scientist because this is not a technician's thing which can be done. And I was very happy to have, before at the Rhine-Waal University, Patric was the student's assistant in my lab, and we started right away with this bioengineering, and we had nothing, and therefore he also built up more or less with me together all these lab courses, and therefore I could easily understand why they, from the Radboud or now Max Planck, they immediately used his knowledge to do that. Now, so you're now bacteria, symbiosis, and so on. We will talk about that later because that sounds quite cool. But did you, before studying bioengineering, did you always dream of diving to the bacteria, doing symbiosis? Was that always your dream to become a scientist in microbiology?
I would say maybe it was a dream to become a scientist, but it wasn't really a plan. So I didn't quite know how to become a scientist, I would say. And it was just a lucky coincidence that I slipped into it, and I like it a lot. Because honestly, like this kind of job is like a big playground. It's so cool that you can just follow your interests. And of course, you have to work also for your supervisors to make everyone happy. But it's really cool to really feed your curiosity in that way. So I couldn't think of a better job right now.
I think it's a good description. Being a scientist means being curious all over your life. You like to learn something new, and this makes you happy. I think that's very important. But did you do the same before? Did you right away start after school with...
No, so I started out to study mechanical engineering and chemistry in Aachen, and then, I dropped out also, worked a bunch of stupid jobs, and then I at some point decided, okay, now maybe it's a good point in life to start over, and then I also took the degree here very seriously, invested a lot of work, and that was really a good decision.
Yeah, I think it's no secret if I say that Patric was certainly one of the best students we had in this year. But I think it's also interesting that it's careers are. It's not always that you have to already think if you are 5 years old that you want to become a scientist later on and work in with microbes, and then you get that. This is very rare, so it's quite usual that the career paths are a bit more unstructured at the beginning, but then if you want to go somewhere, then you just take it and go there. So I think this is a very important point. Now, you started already, but could you nevertheless just briefly explain? By the way, I realized that we plan to have a place with shadow. Now the shadow is a bit disappearing. So if on the video someone sees a drop of sweat, this is just because of the temperatures. Just could you summarize again the research fields you work in? So it's symbiosis, we already learned. So what's going on there?
What's going on there is that it's a really intricate relationship between bacteria and the host. And it's interesting for a lot of different perspectives. Of course, the medical field, how certain bacteria can survive in host cells. That's knowledge we generate there. But also it's interesting to understand our own evolutionary path. I mean, we have bacteria like mitochondria in all of our cells, which help us, but this is a process which has been going on for much longer. So the symbioses we are working on, they're younger than the acquisition of mitochondria. And so it allows a different viewpoint, a different time point of how such thing can form, for example. And my field in particular is metabolomics. So I'm not looking so much at the genomic or transcriptomic side. I'm looking at small molecules. Small molecules means like below 1500 Dalton. And what I try to figure out is which of those molecules get exchanged between symbiont and host.
Okay, and what again were these organisms? You mentioned it at the very beginning, but these vertebrates and bacteria, what exactly were these organisms you were looking about, and why do they do that?
We work in our department on different organisms. So I'm working on deep sea mussels, Bacterial mussels, which are found in the deep sea. They are very closely related to blue mussels, so Miesmuscheln, which we all eat. gutless
And what do the
oligochaetes.
bacteria do there in this symbiotic relationship? Exactly?
So they oxidize substrates, reduce compounds from the environment, such as sulfide, hydrogen, carbon monoxide, or methane. And the energy they generate is shared with the host.
Okay. Do you need to dive so deep to find symbiosis between bacteria and other organisms?
No, of course, we have it on land. Insect symbiosis, a very vivid field of research. But actually, we found those kind of symbiosis first in the deep sea, and everyone was surprised. And then when we looked, we also found it in shallow waters. For example, in the Mediterranean, we have these gutless oligochaetes, which are worms, which don't have a mouth or a gut or an anus, so they can't eat. They're just packed full of bacteria, which do the same purpose as a digestive tract.
But isn't it because it's also cool? This kind of symbiosis. I think of you have a big ship where you then go out for 4 weeks, take some samples, have fun. Isn't that a bit the marine biology?
It is, yeah, for sure.
Okay, that's
so,
good that you confess
so far,
it.
I wasn't fortunate enough to get on a cruise ship yet, like a research cruise. But I've been to Elba 2 times. I've been to Sylt. I've been to Spiekeroog. So field work is definitely something really nice. It's exhausting. You're not, stopping work and then having a cocktail at most days. You're really working 24/7 if the samples are coming. It's a lot of work, but it's a privilege definitely, and it's very enjoyable to go into the field. Yeah.
Yeah, so Patric, it's amazing now. I think that all the viewers now want to become environmental biologists and look for symbiosis in bacteria, which is, of course, a fascinating field. I'm very happy that I had you here back in Kleve. So thanks a lot.
Thanks for having me.
Yeah, and to all the listeners also, thank you very much for your attention. I think this was also a very fascinating field. And you see, he's a first-year bioengineering student, and just after having the Bachelor in Bioengineering, going to become a staff scientist at the Max Planck. That's not too bad. It's with respect to evolution of a bioengineer. So also to you, thank you a lot, and hope to see and hear you again next time.